All right, let's talk about the Benedict's test for reducing sugars. It's funny, most students actually lose easy marks on this topic. You might be thinking you already know the sequence, right? You add the reagent, you heat it up, and you watch it turn that classic brick red. Simple. Well, actually, scratch that. The Cambridge AS and A-Level Biology 9700 exam isn't just testing if you can memorize a colorful reaction. No, it's testing if you can really think like a scientist under pressure. So in this explainer, we're going to see exactly what the examiners are actually looking for, and why your standard, run-of-the-mill revision is probably leaving absolutely crucial marks on the table. Let's dive right in.
Okay, let's expose a cold, hard mistake right out of the gate. You might do a calculation that looks mathematically completely fine, but it can actually be deeply flawed. Why? Because it's entirely missing its units. Contrast that with a top band calculation. That one includes perfectly correct notation and units at every single step of the way. It's crazy, but this tiny, seemingly innocent missing unit error costs students easy marks before they even fully realize what topic they're dealing with. You can know the biology inside and out, but honestly, if you drop your units, you drop your grade. It's that simple.
And that really brings us to a critical question, one that's actually posed directly by senior examiners. Why do students find evaluating evidence and drawing conclusions so incredibly hard? When it comes to the Benedict's test, you aren't just saying, hey, sugar is present. You're often asked to evaluate the actual concentration of that sugar, using colorimetry, analyzing absorbance data, and then concluding whether your evidence supports a specific hypothesis. Ordinary flashcard-style revision, it completely fails here because it just doesn't prepare you to evaluate unfamiliar evidence on the fly. To understand what they want, we have to look at the 3 specific assessment objectives Cambridge targets for this topic. Notice that knowledge and understanding is literally only the first part. The real challenge, the place where the grade boundaries are actually decided, sits right there in handling and evaluating information and your experimental skills. The top-performing students, the ones really locking down those top bands, they don't just memorize facts. They exhibit a fully connected conceptual understanding. They can take the core principles of the Benedict's test and transfer them to entirely unfamiliar, complex experimental context without panicking.
So what's the secret to getting there? Cross-topic connections. And look, this isn't just some helpful tip. This is a direct insight pulled right from the principal examiner reports. The theoretical architecture of the Benedict's test relies heavily on precise definitions and causal relationships. If you can connect the principles of reducing sugars here in topic 2.1 to, let's say, enzyme kinetics or membrane transport in later topics, examiners immediately recognize you as a top-tier candidate. You're showing them you get the big picture. Now, stick with me, because in just a moment, I'm going to show you the exact examiner clue that changes your answer completely. And frankly, it'll save you from the absolute most common traps out there. But first, we need to build the framework that actually makes that clue obvious. Because without a system to process the data, even the best clues will just look like extra, confusing text on your exam paper. To effectively process complex exam questions, you really need this 5-part procedural framework. Step 1, identify the key elements in the stimulus material. Step 2, apply the relevant biological principles, like, say, the specific reduction of copper-2 ions. Step 3, execute your analysis with clear, highly logical working. Step 4, verify your results to ensure they actually make scientific sense. And Step 5, present your findings in the exact format the examiner requested. Think of it as a systematic sequence. It prevents panic and pretty much guarantees you hit those mark scheme requirements.
So, how do we put that framework into action? Well, when an exam asks you to deconstruct, your main execution detail must be to extract only the relevant data from the tables and graphs they provide. Just ignore the noise. When you analyze, you have to select the correct analytical framework. And when you present, it is all about using precise scientific terminology and clear notation. Whatever you do, never just say the color changed. You want to say a brick red precipitate formed indicating the reduction of copper-2 sulfate. In these exams, precision is absolutely everything. Let's ground this in some theoretical statistics you're actually going to need when handling data from these experiments. Choosing the right statistical tool, totally non-negotiable. If your Benedict's test experiment yields 2 sets of continuous data and you need to compare their averages, you're going to use the t-test. If you're dealing with categorical data, like say counting the frequency of different color outcomes, you use chi-squared. And if you're looking for a relationship between 2 variables, maybe sugar concentration and light absorbance, you use a correlation test. Selecting the wrong test instantly caps your marks, so you got to get this right.
Which brings us to the magic number 3. Why 3? Because there are 3 levels of higher-order assessment objectives you absolutely must climb: descriptive, analytical, and evaluative. Describing the data is basic.
Analyzing the data is a step up, but evaluating the data, that is exactly where you secure the highest grades. Evaluation demands that you weigh alternatives, question the validity of the experimental design itself, and fiercely justify your conclusions using the evidence provided to you.
Okay, listen up, because you really need to be aware of this massive examiner alert. Students lose marks by not showing their working. Even if your final answer is dead wrong, method marks can absolutely save you. This is known as the method mark strategy, and it is a total lifesaver. When you're calculating a serial dilution for a Benedicts standard curve or figuring out a percentage error, showing your clear, step-by-step working is non-negotiable. If you punch the wrong button on your calculator but your method is perfectly written out on the page, the examiner can still award you almost all the marks. Seriously, do not do the math in your head.
All right, payoff time. Remember that promise I made to you a few minutes ago? I told you I'd reveal the examiner clue that changes everything. Well, here are the top 3 mistakes pulled straight from the principal examiner reports. Number 1, forgetting units in calculations. Number 2, forgetting to double-check units and terminology against what the specific question asked. And number 3, giving generic textbook answers that aren't specific to the unique experimental context given in the paper. If you actively scan your paper for these 3 traps, I promise you, you're already way ahead of 80% of the candidates taking the exam.
Now, to make sure you never stumble on the basics while you're trying to be super analytical, here's a fast, practical quick win. Create your own mnemonic using the first letters of the key terms for the Benedicts procedure. For example, reagent, heat, precipitate, color. You can make up a silly sentence like, really hot pancakes colorfully, that you'll easily remember under pressure. When your brain is super stressed during the exam, having a simple memorized sequence frees up your cognitive load. That way, you can focus on the hard data analysis instead of trying to remember if you're supposed to boil the solution or freeze it.
This brilliantly illustrates the final difference between an average paper and a top-scoring paper. An average student just describes the data blindly. They accept it as absolute truth without ever questioning it. But a top-scoring candidate, they actively question assumptions, they point out anomalies in the results, and they justify all their decisions with hard evidence from the text. They deconstruct, solve, and evaluate systematically. They aren't just acting like a student reading a paper, they're acting like a scientist peer-reviewing a study. That's the mindset you need.
But wait, here is the ultimate trap. You could know the Benedicts test perfectly. You could show all your working. You could even remember all your units. And yet, you can still lose the majority of the marks. Why? Well, the hidden cause of losing these marks is fundamentally misunderstanding the command words. If the exam specifically says explain and you merely describe, your brilliant biological knowledge scores as 0. Knowing the biology just isn't enough if you misread what the examiner is actually commanding you to do with it.
So, what's the solution? How do you decode these command words so you never fall into this trap? Because let's be real, if you can't distinguish between explain and describe, does your biological knowledge even matter in the exam? Understanding these command words is absolutely the mandatory next step to locking in your high grade. Make sure to subscribe to our YouTube channel, Ace the Exams, at acetheexams_IGCSE, to excel in your Cambridge examinations. And I will see you in that next explainer to decode those vital command words. Keep focused and keep learning.